Namaste students. I am Reverend Dr. Aziz Nurul Khan from the Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapeeth, University Jaipur. Today I am going to discuss about the topic Phosphoketolase pathway in the microorganism. What is the Phosphoketolase pathway in the microorganism? And this topic belongs to the subject fundamentals of microbiology in the second semester and the BSc first second semester. The phosphoketolase pathway is distinguished by the key cleavage enzyme phosphoketolase. Means the enzyme phosphoketolase takes part in this pathway, and this enzyme cleaves pentose phosphate into glyceraldehyde 3-phosphate and acetyl phosphate. So that's why we know it as a phosphatidylase pathway, and it is also known as the retrolactic pathway. As a fermentation pathway, it is employed mainly by the retrolactic acid bacteria, which includes some species of the lactobacillus and leuconostoc. So this pathway is found in the lactobacillus and leuconostoc families. Uh, in heterolactic fermentation, the end product is ethanol, and similarly, the end product is carbon dioxide, and the additional product is the lactic acid. So, the end product is ethanol and CO2 in addition to the lactic acid. And in this reaction, glucose is first metabolized to pyruvate, acetic acid, and CO2 by pentose phosphate pathway. Okay, the glucose is metabolized pyruvate. Okay, then acetic acid and then carbon dioxide by the P2P pathway, pentose phosphate pathway. And pyruvate is then reduced to lactic acid, whereas acetic acid is reduced to ethanol and carbon dioxide. Okay. So now go to the steps of this pathway. What are the different steps of phosphatidylase pathway? In this pathway, first glucose phosphate. Okay. Glucose phosphate is oxidized to glucose 6 phosphogluconic acid. Glucose 6 phosphate, that is the pathway in the glycolysis. So glucose is converted into the glucose 6 phosphate, and this glucose phosphate is oxidized to 6 phosphogluconic acid. That is the first step, which becomes oxidized. Okay, it becomes oxidized and decarboxylated to form pentose phosphate. So in the first step, the product is pentose phosphate. Okay, now go to the second step. Unlike the middle pathway, NAD mediated oxidation takes place. Okay, before the cleavage of the substrate being utilized. So pentose phosphate is subsequently cleaved to glyceraldehyde 3-phosphate. Pentose phosphate is subsequently cleaved to glyceraldehyde 3-phosphate and acetyl phosphate. And then GAP is converted. GAP is converted to lactic acid by the same enzyme as in the EM pathway. And this branch of pathway contains oxidation coupled with reduction, while 2 ATP are produced by substrate level phosphorylation. Acetylcholine acetyl phosphate is then reduced into two steps to ethanol, which balances the two oxidation before the cleavage, but it does not yield ATP. The overall reaction is glucose is converted into the lactic acid, and then ethanol is produced as carbon dioxide with the gain of the 1 ATP. But the efficiency of this pathway is half about the EM pathway. Okay. So we must also repeat the steps here. First, glucose phosphate is oxidized to 6-phosphogluconic acid. Glucose phosphate. First step is the glucose phosphate is oxidized to 6-phosphogluconic acid. Okay. Then 6-phosphogluconic acid becomes oxidized and it decarboxylated form pentose phosphate. Then pentose phosphate is subsequently cleaved to glyceraldehyde 3-phosphate and acetyl phosphate. The glyceraldehyde 3-phosphate is converted to lactic acid by the same enzyme that was used in the EM pathway. The intermediate product formed are 1,3-diphosphoglyceric acid, 3-phosphoglyceric acid, and phosphoenol pyruvate and pyruvic acid. Okay. Then this branch of pathway contains oxidation coupled to a reduction, while 2 ATP are produced by the substrate level phosphorylation. The acetyl phosphate is reduced in two steps to ethanol through acetaldehyde, which balances the two oxidation before the cleavage but does not build ATP. So what is the overall reaction? Glucose is converted into the one lactic acid, one ethanol molecule, and one carbon dioxide with the gain of the 1 ATP. Okay. Now talk about the application of the phosphoketolase pathway. Heterolactic species of bacteria are occasionally used in the fermentation industry. For example, caffeine, it is a type of fermented milk leather and it's produced by produced using a heterolactic bacteria that is the lactobacillus species. It utilizes this pathway. So this pathway, phosphoketolase pathway, is utilized by some specific bacteria which are industrially very important, like lactobacillus species. Like some sauerkraut fermentation use leuconostope. It is also a heterolactic bacteria which complete which utilize this pathway for the fermentation. So this is all about the phosphoketolase pathway. So I hope the students have better understood all these things. So what kind of questions which can be asked from this lecture? The question which can arise from this lecture: explain phosphoketolase pathway, explain the enzymes which are involved in the phosphoketolase pathway, what are the different steps in the phosphoketolase pathway, what is the applications of the phosphoketolase pathway. So these questions can be answered by the students very well. So my lecture is over here. If you have any query, you can ask me. So thank you very much.